guys, this is Zoologist AK and I'm here with another topic, that is genetic dissection to investigate gene action. From this topic I have got three questions in different question papers that I pick up from previous year question paper of CSIR net. So I think this is an important topic to discuss. Actually this is nothing but a mechanism or a procedure to investigate gene action, due to which we get to know by mutation that which gene has what function. So let's understand this first, then I will discuss the questions. So before understanding this genetic dissection that investigate gene action, we need to understand the beadle experiment, beadle and total experiment, or beadle and total hypothesis, that is one gene, one enzyme hypothesis. In 1941, Beadle and Tatum built on this connection between genes and metabolic pathway. Their research led to the one gene and one enzyme hypothesis. It states that each of the enzyme that act in a biochemical pathway is encoded by a different gene. Beadle and Tatum used the fungus neurospora crassa, a mold, for their studies. Actually, this neurospora was prototrophic. What does it mean by prototrophic? It means it could synthesize its own amino acid when grown on a minimal medium. Minimal medium means a medium which lacked most nutrient except for a few minerals. For example, it includes some simple sugars or one vitamin, example biotin. And they also knew that by exposing neurospora spores, as neurospora is a mold, so by exposing neurospora spores to X-rays, they could randomly damage its DNA. Means that X-rays damage the DNA of these neurospora spores that create mutation in the gene of neurospora. Each different spore exposed to X-rays potentially contain a mutation in a different gene. After genetically screening many spores for growth, most appear to still be prototrophic. Prototrophic means, as I have discussed, that they could synthesize its own amino acid when grown on a minimal medium and still be able to grow on minimum medium. But some spores had mutations that changed them into auxotropic strain. What does it mean by auxotropic strain? It means that they could no longer grow on minimum medium but did grow on complete medium supplement with nutrients. In fact, some auxotropic mutations could grow on minimum medium with only one or single nutrient supplement, such as arginine. So by this experiment, they give us this mechanism that we are going to discuss, that genetic dissection that investigate gene action, because in this neurospora case, same Beadle and Tatum also did the same experiment. They also get to know which gene has which function and which gene has the capacity to grow in the medium and another one has not. I am talking about Beadle and total experiment, then a question arises why I am talking here about Beadle and total experiment. Because this experiment demonstrates the utility of screening for genetic mutants to investigate a biological process. And this biological process is nothing but genetic analysis. For example, Sir and Horowitz in 1944 tested the ability of different amino acids to rescue auxotropic strains, and these amino acids were the intermediate metabolites that take part in the arginine biosynthesis. Because as we know, neurospora is a mold and it has two cycles, sexual and asexual cycle of reproduction, and it does produce spores. So Horowitz does nothing but grow the mold spore in different test tubes that consist of different media or called minimum media. He want to check the ability of spore to grow in the minimal medium that consists different types of amino acid as a supplement. For example, ornithine, citrulline and arginine. Before understanding this, let us understand the biosynthetic pathway of arginine to understand the table in clear way. This picture here shows us the biosynthetic pathway of arginine, that is a metabolite and that is used by neurospora, and minimal medium that consists this arginine neurospora spores grow in the normal amount. And these arginine biosynthetic pathway consists two intermediate metabolite, that is ornithine and citrulline, and different types of gene activate to produce the arginine in the intermediate steps. As we can see here, the precursor is converted to ornithine by an a product of gene A, that is the enzyme A, and the product of gene B, that is the enzyme B, convert ornithine into citrulline, and then this citrulline is converted to arginine with a product of gene C, that is the enzyme C. So if there is a mutation in gene A or in GC, it will not able to produce the arginine, and if there will be the wild type gene A and gene C, and if there will be the mutation in the gene B, then also there will be the no biosynthesis of arginine. So this is a complementary pathway because it consists intermediate steps. So in any metabolites or in any biological process that consists intermediate steps or intermediate metabolites, a step of reaction that produce the end product, for example the arginine here, that any mutation in a particular gene that take part in the biosynthetic pathway inhibit the synthesis of the end product. So the Horowitz and Sub wanted to know the mutation, which mutation result in the ungrown spore in a particular minimal medium with a particular amino acid that consists that particular amino acid as a supplement. As we can see here, this single mutagenized spore, mutagenesis is produced by X-rays, and this mutagenized spore is transferred to another test tube and incubate in a different types of media that consist different types of amino acid as a supplement. As we can see here, that spores are tested here for their ability to grow on different types of media, because spores of this particular colony are able to grow only on complete medium. As we can see, these CM denote here the complete medium, and these spores grow here. These represent the growth of the spores. So these spore represent here that these mutagenized spores only grow in the complete medium, but when we supply these spore a medium that consists sugar or a biotin that is called as minimal medium, these spores doesn't grow. When this minimal medium is supplied with different types of amino acid as a supplement like alanine, leucine, cytosine, methionine, tryptophan, valine and tyrosine, these spores doesn't grow. But when this minimal medium is supplied with an arginine as a supplement, then this particular spore does grow, and they are considered arginine auxotropes, and we infer that they have a mutation in a gene in the arginine biosynthetic pathway. This type of screen is repeated many times to identify other mutants in the arginine pathway and in other pathways. So Biddle and Totem experiment are not important only for its conceptual advantage in understanding genes, but also because they demonstrate the utility of screening for genetic mutants to investigate a biological process or genetic analysis. And Biddle and Totem results were useful to investigate biological process, especially the metabolic pathways that produce amino acid. For example, Sub and Horowitz in 1944 tested the ability of amino acid to rescue auxotrophic strains. As we can see here in this chart, that when wild type is grown on minimal medium, it was able to grow because it has the capacity to synthesize different types of amino acid for its metabolic pathways, and it does grow in the minimal medium that was supplied with ornithine as a supplement and citrulline as a supplement and arginine as a supplement. But if there is a mutant one that consists some types of mutation, but we doesn't know which gene is mutant is here, so it doesn't grow in the medium that is consist minimum medium or consist sugar or biotin only, and it grew in the ornithine, citrulline and arginine. It means that mutant 1 has the mutation in the enzyme 1 that convert precursor to ornithine. Because this step is missing, because it couldn't grow in the minimal medium but it grow when ornithine is present, because enzyme 2 is function, enzyme 3 is function, so it can synthesize ornithine to citrulline and citrulline to arginine and can complete its metabolic pathway. And mutant 2 were unable to grow in the minimal medium, and the minimal medium that consists ornithine as a supplement. It means it consists a mutation in the enzyme 2, because due to non-functional uh, non-functional presence of enzyme 2, it is unable to convert the precursor into ornithine and then ornithine to citrulline, but it does grow in the citrulline and arginine because it has the ability to convert citrulline into arginine, because its enzyme third is active in the process and arginine is the end product, so it is able to grow in the arginine. And mutant third didn't grow in the minimum medium and not grow in the minimum medium that consists of ornithine and citrulline as a supplement, because enzyme third was mutant here, because this enzyme mutation unable to convert citrulline into arginine, and it could not convert the precursor into ornithine. If does it convert precursor into ornithine and ornithine into citrulline, but it this spore is unable to convert this citrulline into arginine, due to which it couldn't grow in all the three minimal medium or medium with ornithine and citrulline as a supplement, but it could grow on the arginine because it is the end product of the biosynthetic pathway. So by analyzing the mutation or the ability of the neurospora spore, that on which medium it could grow, we get to know that there is the mutation in particular gene that take part in the steps of the biosynthetic pathway of arginine. Or we can say that if the progeny of a mutagenized spore could grow on minimal medium only when it was supplemented with arginine, then the auxotrope must bear a mutation in the arginine biosynthetic pathway and was called as arginine less strain or a arginine less strain or ARG negative. This Beadle and Tatum experiment opened the way to investigate of the roles of individual gene mutation in biosynthetic pathway. And these investigations begin with three assumptions about biosynthetic pathway. And these assumptions were: the biosynthetic pathway consists of sequential steps, and completion of one step generate the substrate for next step in the pathway. As we know that if citrulline was formed, then after citrulline arginine will form, and if ornithine is formed, then after ornithine citrulline will form. So these completion of one step generate the substrate for next step in the pathway, and completion of every step is necessary for production of end product. If there is a no, there will be the synthesis of the ornithine from the precursor, then citrulline, and then arginine, then the biosynthetic pathway will complete. And if there will be the no synthesis of the ornithine from the precursor and then citrulline, then there will be the incompletion of the arginine biosynthetic pathway. So completion of every step is necessary for the production of end product of the pathway. These assumptions support the conclusion that wild type strains are able to complete each pathway step, that mutants strains are unable to complete a pathway because one or more pathway steps are blocked by mutations. So genetic dissection is an experimental approach that determine the number of intermediate steps within a biosynthetic pathway. How much intermediate steps are there? As we discussed here about the arginine biosynthetic pathway, so there are the precursor, then ornithine, then citrulline, and then arginine. So here are the three steps or three enzymes basically that are required for the completion of biosynthetic pathway. So we get to know about intermediate steps and determine the order of steps in the pathway. What is the order? What is the first substrate, and then second substrate, and then third substrate? For example, here precursor, then first is the ornithine, second is the citrulline, and last one is the arginine, and identify the step that is affected by each mutation as we have discussed in this chart. And the genetic dissection to investigate gene action, we need to understand an example of biosynthesis pathway of phenylalanine in case of neurospora, that is mold, and the question I will discuss in the next video. Let us understand this example first. This picture here denote the wild type and mutant strains that give us information about the mutations in a particular enzyme that involved in the biosynthetic pathway of phenylalanine. And by studying this mutant, we need to identify the correct order of steps that are involved in the biosynthesis pathway of phenylalanine. As we can see here, from wild type, it has the capability to grow in any types of supplement because it could convert any type of substrate into final product, that is phenylalanine. But in mutant first, we can see here that it has it couldn't grow in the phenylpyrrate, prephenate, and chorismate, but it has the capability to grow in phenylalanine. Plus sign here represent the ability to grow, and negative sign represent here inability to grow. So we can see in mutant type, this P represent here precursor, and A substrate, first substrate, B substrate, second substrate, C substrate, third substrate, and the final product phenylalanine. So first, second, third, fourth represent here enzymes. So we can see here that this mutant first has inability to grow in any type of substrate except phenylalanine, that is the end product. It means that there is a mutation in enzyme fourth, because if these enzymes are functional and wild type, but these product of these enzymes are unable to convert into the end product, that is phenylalanine, due to the absence of enzyme or absence of functional fourth enzymes, or presence of this inactive form of fourth enzymes, due to which these substrate are unable to convert into phenylalanine, due to which this mutant has incapability to grow into phenylpyrrate, prephenate, and chorismate. And then if I talked about mutant 2, and then it has the capability to grow in phenylpyrrate, prephenate, but it could not grow in the chorismate, but it could grow on the phenylalanine. It means that there is a mutation in the type second enzyme that convert these first substrate into second substrate. So it could not grow in the presence of this first substrate. So it could not grow only in the chorismate presence. So it means that A substrate is the chorismate, and because this chorismate is unable to convert in the final product, that is the phenylalanine, but this mutant 2 has the capability to grow in the substrate B, substrate C, that is either phenylpyrrate or prephenate. We don't know yet by this only mutant 2 that we going to clear in the mutant 3. So if we look at the mutant third, that we could get that this mutant third has the capability to grow in the phenylpyrrate and phenylalanine, but it could not grow in the chorismate presence or the prephenate. So we can say that there is the mutation in the enzyme third that convert this B substrate into the C, and then due to this mutation, this C substrate inability of formation of the C substrate, so there is the no formation of phenylalanine, that is the end product. So from this we can see it could not grow in the presence of this B substrate, and this B substrate is the prephenate, because these could not grow in the prephenate presence, as we get to know that chorismate is the A substrate, first substrate. So chorismate is the first substrate, prephenate is the second substrate, and C substrate, third substrate, is the phenylpyrrolate, and the last substrate is the phenylalanine, and P represent here the precursor. So the correct order of metabolic intermediate that take part in the phenylalanine biosynthetic pathways are chorismate, prephenate, and phenylpyrrolate and phenylalanine. These are the correct order of steps. So if you like, then please subscribe, and if you have any query, then write in the comment box given below. Thank you so much for watching my videos.